Sierra.
And the enzyme we chose to study is called thrombin, and it's a bloody cool protease.
So thrombin is a serine protease, which means in its active site it contains a nucleophilic residue of a serine. In any case of a cut or a lesion that causes a vascular injury, aka you're bleeding, it triggers a cascade response of thrombin. Here, thrombin then acts to cut and cleave fibrinogen into fibrin, which acts as an anchor for platelets in the clotting process. So while thrombin can act as a coagulant, it can also act as an anticoagulant, which means it's going to inhibit clotting rather than promote it. So in that pathway, it's known as a slow pathway. Instead of cleaving fibrinogen into fibrin, thrombin actually acts to cleave this other protein called protein C, which acts as a negative inhibitor in the feedback loop of the thrombin process. So it decreases the activity and the production of thrombin so that you don't clot when you don't need to be clotting. So thrombin can further be activated or regulated by ions, specifically sodium ions, which Tara's going to talk a little bit more about in a second to kind of explain the fast and slow forms that you see here. Because in that slow form pathway, you have thrombomodulin, which acts as a regulatory effect as well as the sodium effect on the regulation of thrombin production. In the procoagulation process, though, thrombin, like I said earlier, does cleave fibrinogen into fibrin, but it also cleaves another factor called factor V, which acts as a positive feedback protein in the thrombin. So it actually acts to cleave prothrombin into the active thrombin form.
So I'm going to talk a little bit about thrombin's structure. Basically, when they crystallized the protein, they found two major things. They found the catalytic triad, histidine, serine, and aspartic acid, and that was nestled in this little pocket that was the active site. And they found exocytes, specifically 1 and 2, which are regions composed of positively charged amino acid residues, and their job is to essentially mediate the recognition of substrates, inhibitors, receptors, foreign objects, what have you. And Sierra mentioned how sodium influences the protein. Sodium actually can create conformational changes in the protein, and at physiological pH, the protein exists at 3 different forms. The free form, E, is the low activity conformation. It activates protein C anticoagulant pathway. The bound form, ENA, is the high activity conformation, and it cleaves procoagulant substrates more rapidly. And then the equilibrium form, E, in which Na is unable to bind. In this way, sodium acts as an allosteric regulator for the activity of thrombin and basically creates a balance in the clotting. And here you'll see that we have two cool structures of our protein. We have this globular thing right here where you can see the exocytes a lot better. Exocyte 2, exocyte 1, and the active site pocket, and then over there by Sierra, you can see the domains and the active site residues. So thrombin at the gene level, the F2 gene is the gene that codes for thrombin. It is called coagulation factor 2. It is located on the short arm of chromosome 11 at position 11.2, and I've included a graphic of chromosome 11 for you to be able to visualize that. And as with any gene, F2 is susceptible to mutations. The two main ones for thrombin is a deficiency or thrombophilia, so essentially too little thrombin or too much thrombin. The deficiency can give rise from 50 mutations that usually change one singular amino acid in the protein sequence. Depending on what amino acid is changed, the conditions can be mild to drastic. So essentially, you can not feel the effects, or you can bleed out and die because you don't clot. The thrombophilia side of it is the opposite. The mutation changes one DNA nucleotide, specifically guanine, to adenine at position 2,0,2,1,0, and it leads to an overactive form of the F2 gene, which creates too much prothrombin that leads to unnecessary clotting when you don't need to clot, and that can cut off blood flow and oxygen circulation to important parts of the body, such as tissues, organs, and limbs. There are 9 different mRNA sequences that exist for the F2 gene. The one here I've included is basically the proper accepted one. It produces 622 amino acids, and the other ones are of varying protein integrity. Some are better than others, and some of the mRNA sequences don't code at all. So now we're going to segue into our paper, the role of thrombin exocytes 1 and 2 in the activation of human coagulation factor 5.
So the major goal of this paper was to study the specific exocytes on thrombin, so those exocytes 1 and 2 that we've talked about, and they wanted to study the specificity of those exocytes on the cleavage sites of factor 5, which I mentioned earlier is a positive feedback protein in the prothrombin into active thrombin cascade process. So pretty much cleave prothrombin via factor 5 so you get more clotting. So their main goal, like I said, was to study those two exocytes. Because the coagulation cascade is so complicated and there's so many proteins involved, this study specifically focused on factor 5 rather than kind of all 13 proteins that can be involved in this. So there was a number of methods that they had to use to determine protein concentrations and various variants that had to be made for this test to be able to happen. So the first thing that they did was they measured the protein concentration by using the VCA protein assay, which we've talked a little bit about in class. And they did that initially so that they knew how much factor 5 was needed for each sample and how much thrombin was necessary for each sample. So for the thrombin, they specifically use another substrate called chromogenic substrate S2238, which fits neatly into the active site of thrombin so that they could further determine the concentration levels before those samples were used. And then for the mutant variant types that they had to make for the factor 5, instead of altering thrombin's active sites itself, or its exocytes itself, they actually made mutants of factor 5. So in factor 5, they changed around 3 specific arginines, arginine 709, arginine 1018, and arginine 1554. 1545, pardon me. So at those 3 arginines, they made 6 variant forms that had varying residues at those 3 specific points to study how thrombin's exocytes would cleave or not cleave at those sites. So that's what the variants were they used. And then after all their samples and tests were run, they ran an ELISA assay. So this is after thrombin had been introduced into the samples. They measured the expression levels of active factor 5 by using that ELISA assay. And they did that by creating a steady state curve, which we'll see in just a second. So since there are 2 exocytes on thrombin, they had to study one at a time. And to do that, they use these things called aptamers. Aptamers are pretty much like inhibitors for the exocytes rather than the active site. So aptamer 1 was used to block exocyte 1 so that they could study exocyte 2. And then aptamer 2 blocked exocyte 2 so they could study exocyte 1. So after determining the activation levels of factor 5 using thrombin, they continued on their studies a little bit into the use and inhibition of heparin on thrombin. So heparin is a known inhibitor of coagulation. So you might have heard of it in medicine as the blood thinner. So pretty much it is known to interact with thrombin's exocyte 2, but they're not really sure how. So this was kind of their first dip into seeing how the inhibition of heparin is actually affecting exocyte 2. So they wanted to start off with that, and they did that by testing the catalyzed thrombin activation levels of factor 5. Finally, after all of these tests, most of them were kind of inconclusive, which we'll see in a second. So they ran STS page gels with samples at varying time variants over an hour. So each sample was placed in a well, and then they ran the gel to see at what time point the factor 5 was actually being activated. So this was their very initial test. They wanted to see if increasing the amounts of thrombin would increase the amount of thrombin being produced, aka more factor 5 is producing more thrombin. So what they did was, up here on this top line, they have their wild type, which is that RRR variant, or not variant, it's just their wild type, pardon me. And then the second line below here was really their big suggestion that the RIR variant, which is that arginine 709 variant, is actually being cleaved by exocyte 1. So that was just kind of their preliminary test, which led them into their following test.
So these 3 graphs are representations of each arginine that is in the wild type, and they just subbed the other arginines with amino acids that wouldn't hurt the sequence. This first graph is these open diamonds right here that are following the wild type graph. This is exosite 2 blocked, and it shows successful activation of arginine and cleaving of arginine 709, which means, or which stated to the researchers that exosite 1 was involved in the cleavage of arginine 709, exosite 2 wasn't really. And then we're focused on this graph over here, which unfortunately didn't really tell our researchers anything. Down here, these two lines are where aptamer 1 and aptamer 2 were used to block exosite 1 and exosite 2, and there was pretty substantial inhibition, as you can see, and then there's a line on the very bottom that may be difficult to see, but that is complete inhibition, no activity at all whatsoever for the enzyme, with both exosites blocked. So the researchers were kind of like, okay, what do we do with this? Because we have some sort of significant data here, how else can we visualize it? And that's when they used SDS Page to visualize what was going on. We're going to focus on these two gels right here. If you can look, in aptamer 2, the light chain development, which is dictated by arginine 1545, is down here. And the light chain doesn't really start developing until around 25 minutes, and that's with exosite 2 blocked. Up here in aptamer 1, with exosite 1 blocked, exosite 2 functioning, the light chain starts to develop around 15 minutes as opposed to around 25 minutes. And that's kind of how our researchers drew the conclusion that, hey, exosite 2 must have something to do with the light chain development in our thrombin and the cleavage of arginine 1545.
So after looking at the exosites and their specificity on factor 5, like I said earlier, they wanted to test the inhibition effects of heparin on thrombin. Thereby, they looked at the activation of factor 5 to study that. So this is going to be a little confusing because of this inset, but I promise it's not that difficult. So this bottom, so your x-axis, your bottom axis, is just increasing concentrations of heparin, units per milliliter. And then your y-axis is just that amount or concentration of activated factor 5. So here you can see that, as expected, at high amounts of heparin or high concentrations of heparin, you have very low activation of factor 5, which is expected since it is known as a blood thinner. However, what they did notice on this inset is that it's actually just this first part of the graph. It's kind of blown up for you. But what they saw is that at those very small concentrations of heparin, so 0.2 to 5 units per milliliter, there's actually an increase in the rate and an increase in concentration for that factor 5 activation. So they found that really interesting, and which is what led them into their next study of heparin. So here, on the top line, those closed circles represent when heparin is present, and these open circles represent when heparin was absent. So that was really interesting because when heparin was absent, they had lower factor 5 concentrations over time, which was very interesting because this is representing a 10 units per microliter concentration of heparin that they used. So when it's absent, they actually had a very low activation. So this kind of suggested to them that they want to do more studies to see how heparin is affecting the exosite 2 on thrombin, because they suggest or propose that the heparin might actually be causing a conformational change in exosite 2, which is causing a higher likelihood of that coagulation pathway rather than that anticoagulation pathway as is known for heparin. So that's kind of what they intend to do from here on out with the heparin studies.
So for the major results, they basically found two solid pieces of information. They said that exosite 1 plays a critical role in the cleavage of arginine 709, backed up by SDS Page, and exosite 2 is involved in the cleavage of arginine 1545, but not 709 or 1018, also backed up by SDS Page. And essentially what they said was, okay, aptamer 2, or exosite 2, must be linked to the cleaving of arginine 1545 and light chain formation for factor 5, and exosite 1 must be linked to arginine 709 and heavy chain formation for factor 5. And future studies needed to be conducted for heparin.